Hi, everyone. Welcome to our second day of the conference. Thank you all for making the first day so interesting and productive. Today, we begin with a talk by Professor Mathew Leibold. Dr. Leibold is a professor in the Department of Biology, University of Florida in the United States. He has significantly contributed to the niche theory and the development of the metacommunity concept, among others. His works on metacommunity ecology and evolving metacommunities, the niche concept, and on the role of spatial dynamics in aquatic food webs have revolutionized the field of modern community ecology. He's also interested in understanding how evolutionary processes influence biodiversity patterns across spatial scales. To do so, he investigates the link between metapopulation and metacommunity dynamics, using mainly Daphnia as a model species and ponds or shallow lakes as model systems. His research often combines several approaches, such as field observations, indoor and outdoor experiments, as well as modeling. Dr. Leibold, thank you so much for joining us today. The virtual floor is yours. I'll be in the background organizing the questions. And just as a reminder to the participants, we'll keep track only of the questions provided in the Q&A tab, but not in the chat. Thank you again for joining us.
Okay, I have one question, which is it possible at all to see some people's faces?
You can see my face and Coca's face.
No, I can't see Coca's face.
He can turn on his camera.
Okay, it just helps me, you know, because otherwise I feel very lonely.
Sure.
So, okay, thank you. So thank you for everyone to come. I wish I could see your faces too. There's 109 people. I guess that's tough. But anyway, I thought before we start I thought I'd say something personal. This year, at least for me, has been probably the toughest year in my life. Sebastian Deal says, I'm reading your lips, Matthew. What does that mean? Does that mean that nobody can hear me?
I can hear you just fine.
So can Pedro. Sebastian, it's your problem. Okay, so it's been one of the toughest years. And I think it's important to remember why we are scientists and why we are working on what we work on. And, you know, there's so many other things to focus on, and they are really important. But you know, at the end of the day, we are scientists and we are trying to save the planet. And that's also really, really important. And the other thing that I realized this year is just how important other scientists are, my colleagues and my students in my lab, and everyone in the research community. And so I think the problem for me has highlighted just how much of a community we are. So speaking of which, I'm mostly a community ecologist. That was a good transition, maybe. So mostly I'll be talking about community ecology. I work on lakes and you know, some of them are very shallow like that. That's important, but really it's, you know, mostly I think about community ecology in broad terms. And the other thing that I realized recently, this year especially, is that community ecology, while we weren't looking, has been making incredible developments that I think will change the way we do community ecology and the way we change how we do every type of ecology, including limnology and shallow lakes. So, so one of the things I think about these developments is that they are going to change how our ability to address biodiversity at the real scale at which biodiversity occurs. So what I mean by that is that our older approaches, older experiments, older theories make sense for 2 species, 3, 4, 5, and then, you know, we are not smart enough to extend those ideas very much further. And I think these new developments take us into this new realm of being able to think about dozens, hundreds, maybe thousands of species. So that's what I'll be talking about. The other thing about what I want to say is that my talk is going to be very speculative. I don't have a prescription. So, you know, it's mostly going to be a vision, and I will try to make it as precise as I can, but these methods and ways of thinking are changing so fast that anything I say today will be wrong tomorrow. So I'm mostly hoping to get all of you to think in those terms. Okay, so maybe I will share screen. Hang on, I lost my PowerPoint. Okay. I know what I'm doing, I think. There you go. Okay, are we good?
Yes.
All right. Okay, so the title of my talk is Rethinking Metacommunity Ecology. And what I want to think about here is what is metacommunity ecology? So maybe not all of you have drank the Kool-Aid, as they say in the United States. But metacommunity ecology is the idea that ecological dynamics can occur on multiple scales, maybe more than 2, but I'll just emphasize 2. So we're used to thinking about much of community ecology as being at a local scale. And many of us came into ecology this way. You go into a pond and you see all this wonderful life that is going on with tadpoles and bacteria and volvox and crayfish, and then, you know, there's even fish and insects in here, etc. And that's fascinating. But another way to think about ecology is on a landscape. So here's a photo of tundra ponds in Siberia. And so you can see that these ponds differ from each other. Some of them are clear, some of them are opaque. The ones that are opaque are different colors. Why is that? And why is it so different? And do these ponds affect each other? That's the question of metacommunity ecology, is the idea that there's feedback between what's happening at the local scale and the regional scale. So I got into it very much by chance. I used the word metacommunity and people didn't know what I meant. And so I realized that what people meant wasn't this process, this feedback process, but rather specific ideas about how this feedback occurred, and especially based on the work of Levins and Culver. So I got very lucky because one of my colleagues suggested that I have a working group at NCs in the United States to try to resolve the various ways people think about metacommunities. And so that led to this synthesis, which here has, I think, 14 other authors. So it sounds like it's me, but really it's 15 people. This paper was important because it provided kind of a menu of options for thinking about metacommunities. And that led to, you know, in the subsequent 15 years or so, there's been a huge amount of development in this area. And together with my previous student, John Chase, we tried to summarize these developments in a book that came out a couple years ago. And one of the most intriguing things for me was to realize that the idea of metacommunity ecology related to so many other ideas in ecology that it changed the way we think about these other ideas. So I think of it as a hub for community ecology. Maybe not everyone, maybe somebody would put traits and phylogeny in the hub, but I'm going to put metacommunity in the hub. So That's the way I think about it. So it's useful because it helps us try to understand patterns. And I'm going to tell you a little story about myself that relates to this. So when I was an undergraduate at University of Arizona, I took a graduate course in field ecology from Jim Brown. And what that field ecology course consisted of was sitting around thinking about possible projects and then spending spring break 1 week working really hard to get data. And we did 2 projects, but one of them was studying very small, very shallow ponds in these rock pools in the canyons of Arizona. They're called tinajas. And they kind of look like this. And we just ran up these canyons, and a different person from the class would be in charge of measuring something in these rock pools. I was in charge of measuring insects, counting different insects, because I had taken aquatic entomology the semester before. And so I was the expert, which I was not. But anyway, I was like brown beetle with stripes. Okay, they're present. I found 5 of them. That's what I did. And somebody else did snails. Somebody did, other things. And then after that, we were supposed to do a project analyzing the data, whatever. I thought, well, okay, I'm going to figure out if competition is more important or predation is more important in affecting these species. And the way I thought I would do that is take each species one at a time, try to figure out who its competitors were likely to be and who its predators were likely to be, and measure them and do regressions. So I did that. And the first time I did that, I found that all the correlations were positive. So that was not expected at all. I was like, wait a minute, these are predators and competitors. I'm looking for negative correlations. Oh, wait a minute, some of these ponds are really big and some of them are small. Duh. So fortunately, someone had measured the area of the pond and the depth. So I divided by the volume and looked at it again, and still all the correlations were positive. Oh, maybe they differ in productivity. Well, okay, can I get a handle on that? Fortunately, somebody had taken a picture of the sky and estimated how much light was reaching each pond. So if light was the limiting resource to productivity, I had an estimate of that. So I corrected for that. Still, all the correlations were positive. So, okay, light isn't everything, and we didn't measure the rest. So, okay, what do we do? So I said, well, all the other species, we can get an estimate of the productivity by measuring the abundance of everything. And so, more life is more productive. So I put that in, and I found that when I did that, I had some negative correlations with predators and competitors. Hooray! Yes. Which one is more important? Oh, they're both about the same. Wow, great. And then that was the day that we were supposed to present our findings to the class. I did that. I was the only undergraduate there. I was very shy. I found some negative correlations that kind of, I don't know. And Jim Brown, who, if you have met him, is very enthusiastic. He was just like, oh, this is fantastic. Science, nature, So it's like, just went home and, tried to disappear. But at 2 o'clock in the morning, I was like, there might be some bias here, because I included the species themselves in the estimate of productivity. So that's not 2 separate analyses. I'm correcting for the abundance of the things that I'm going to keep before I study their correlations. So the next day I went to the computer center, fixed that, and I had no correlations. The story I came up with is there's lots of confounding factors: area, productivity, light, etc. And even if you try to fix it, you end up with Nothing. So that was a bit disappointing. But 50 years later, I guess, or almost 50 years later, I think I have an answer. So one of the biggest problems is indirect effects. So we think, oh, competitors, predators, they should have direct effects. Yes, they should, but they can also have indirect effects. And indirect effects means that the net interaction, what we will see in these ponds, is not just due to the direct effects. It's also due to the fact that there are indirect effects. So the predator that we think is really bad could be helping us by wiping out our competitors. So there's a whole bunch of theory on this.
It seems like he froze. Did he freeze to everybody?
Yes, Priscila. Mathew.
I'm back.
Oh, he's back. Yeah.
Does anybody have?
Hopefully that won't happen again. Okay, are we good? Yes?
Yes.
Okay. So who would have thought that that would be a reasonable thing to do? I don't know. Tiffany and I did, I guess. And what these people found is that fish had a negative effect on larval dragonflies, which affected how many adult dragonflies were next to the pond, which affected how many pollinators were near the pond, and that affected the plants. So, you know, these are the kinds of issues that we're thinking about. This study also shows that it's not very easy to figure this out. It can be done, but not necessarily that easy. So if we have simple chains of interactions, we can figure them out. But most biodiversity is not that simple. Here's the simplest food chain or web that I have collected, mostly because it's studied by my very good friend, Tom Miller. And it consists of a fairly simple web. So if one dedicates one's life to figuring out these interactions like Tom did, you know, you can do it. But nature is not that simple. So here's a study by Sonia Kiffy and scientists from Chile, looking at the species interaction webs in the intertidal of Chile. And what you can see is spaghetti. Okay, so, you know, how many lifetimes would it take to figure this one out? I don't know. I don't even want to think about it. It's not worth thinking about because I will be dead by the time we figure this out. But that is the problem that we have. Now, these indirect effects have been studied, and you know, the idea goes back to Darwin's entangled bank and Elton's work describing interactions in webs. The kind of the conclusion that we can come from this is two things. One of them is that a change in anything in the web can affect any other link component through these indirect chains, even though those links might be quite distant, just like the fish and the plants. The other thing is that the net interactions between two species can depend crucially on the presence or absence of any of the other species in the web. Okay, this is a problem that's even bigger because if we are Sonia Kiffy and we try to figure out the interactions in that Chilean intertidal, they will be different if any of those species goes extinct. Okay. So, and this idea was developed by this guy, Bill Schaefer. I'm showing a picture of him here because he died a couple weeks ago. And I took his class in Arizona. It was the hardest class I ever took by far, and I hated him. And he was not a nice guy, but this is one of the most important papers I read. It's not an easy read or anything, but it is an eye-opening paper to read as a community ecologist. And we can see that a little bit in this other work by a guy named Larry Lawler. I don't have a picture of him because he did this when he was an assistant professor, and then he quit ecology and became a furniture maker, I hear. Anyway, he figured out that we can get the net interactions from the Lotka-Volterra equations in a web by inverting the matrix of direct interactions. So it's kind of, you know, oh, isn't that so cool? Wouldn't it be so nice if that was the answer? Like, all we have to do is invert. Hey, no problem. We can do that. And so he did that in this thought experiment with MacArthur's warblers, who, assuming that they compete with each other based on overlap in where they are in trees. And by doing that, he calculated the direct matrix of interactions shown here, in which all the interactions are negative, so shown in yellow. And then he inverted that matrix, and he got this, in which two things. One of them is, but on average, these negative effects are not as negative. And the other one is that some of them become positive. How can that be? And so the explanation for that is, if we look at these two birds here, they're the ones who compete with each other in direct interactions, but are mutualists in indirect interactions. And the real reason is that they both compete with this other species over here. And so by suppressing this other species that they overlap with from different sides, they are mutualists by suppressing their mutual competitor. So that's the kind of idea that we want to account for in these in diverse communities. So we can develop that theory a little bit more and think about competition based on something like overlap. But really, competition is rarely that simple. It's usually in a food web of some sort. And so that means that we should be a little bit careful how we think about models that are just based on competition, like the one that Larry Lawler just showed us, because really they're like this, they're indirect interactions. And MacArthur showed that you could do this under some conditions, and he did it with a little food web model like this: two trophic levels. There's consumers and resources, Lotka-Volterra equations, blah, blah, blah. And then down here, he derived how you get the interaction coefficients and the carrying capacities from these equations. So looks nice, right? We can pretend that these indirect effects are abstracted. We don't have to worry too much. Maybe not. Mark McPeak has a book that should be coming out pretty soon. Maybe it's already out, I don't know. Showing that this works when we have resource competition the way MacArthur set it up, but it doesn't work with the other possible configurations. So, I don't know. But anyway, for right now, this is what we're dealing with. And so, you know, I'm an optimist. I'm a half full kind of person. I look at theories and I go, well, if they're helping us, that's good, even if there are some things to worry and be cautious about. My grad students tell me that the most common thing I say to them is, you do the best you can with what you have. And I thought I got that quote from my dad, but really, apparently it comes from Theodore Roosevelt. And he also added, where you are, whatever. I tried to find pictures of Theodore Roosevelt as an ecologist, and they all look like this. He always has, he's in front of some cliff or some canyon or something like that, but his posture, his position is always exactly the same. So it's kind of interesting. So, so the problem here is that this inverting of the matrix works as long as none of the species go extinct. So if we had those warblers and that black-breasted, whatever, green, the one that was in the middle was extinct, those two birds would still be competing. So it depends on the presence or absence of that one species. So what about those extinctions and colonizations? Well, they relate to community assembly. We have a regional species pool, like in Kefi's web, maybe in a single location in Chile, you don't have all those species. You have just some of them. And that's because there's environmental filters and because the biotic interactions influence the community assembly, so that in different local communities, you have different compositions. And therefore, in this community, the net interactions are going to be different than in this community and in this community. That sounds complicated. So the community assembly theory kind of has these three processes that's important, but there's a lot of stuff that will also be important that can complicate things. So Mark Vellin more recently kind of said, well, let's forget about Volterra, paradigms, and let's think more about fundamental processes. So rather than trying to write these in equations, we can just kind of think of them in the context of community assembly. And his idea was to use this as a parallel to evolutionary theory, in which we have four basic processes. One of them is selection, and we can further dissect that into abiotic and biotic selection. One of them is drift, which in community assembly would be stochastic demography. And I should have put dispersal here, which is similar to gene flow, and speciation, which is similar to mutation. We're not going to talk about speciation because, well, too complicated. But it's what his suggestion points to is that maybe we can use this type of thinking to guide the way we think about community assembly and maybe metacommunities. So the reason I say maybe metacommunities is because metacommunities is a little bit more complicated because it has the feedback, not just community assembly going this way, but also the species pool in the region being influenced by the additive process occurring over all the local community. And the reason I'm titling the talk Rethinking Metacommunity Ecology is because currently much of metacommunity thinking is oriented around these four paradigms that we identified in the 2004 paper. And since then, we found that these are just way too simple. And yeah, it was great. It helped us make progress, but nowadays it's time to be a little bit more inclusive about the ideas that we use. So one of the important things, I think, in community ecology has been the work being done generally in metacommunity ecology. Another thing that's really important is that there's more powerful, comprehensive statistical approaches. And I'm going to focus on joint species distribution models, but I think Pedro is going to try to convince you that there are other alternatives. That's my guess. But anyway, here's one of them developed. Here's a framework for one of them developed by Otso Ovaiskanen over here, building a little bit on the work by Francis Hui before. The idea is that we can have multiple sets of predictors that help us understand the distribution of species among sites. They include environment, spatial and temporal aspects, and species niches here, which means species interactions. And so that's kind of the idea. So can we use these joint species distributions to better understand the processes that I've talked about, and to kind of explore this, we had a working group at IDIV by this cast of characters. This guy right here is Leibold. You'll see him, and Luke later in the week over here. And two other people that were not present in this thing. And the idea was we would develop a process-based model that we knew, and we would look to see what kind of results it would give and analyze them using joint species distribution models, like the one by Ovaisky. And we would see if the variation components, one of the output of the Jsdm, makes sense, and we would see if the parameters also make sense. And we think of this as studying the internal structure of the metacommunity. Maybe that'll be a little bit more clear, but what I want to say, what I mean by the internal structure is that instead of focusing on overall kind of emergent properties like average beta diversity or something like that, we study the contribution of each species to the pattern and of each site to the pattern. So here's kind of the little cartoon of the history that we might think of. We might even start before Burckhardt et al. By thinking of just RDA or PCA analyses, which only focus on environment. Burckhardt et al. And subsequent work by people including Pedro show that we can partition overall variation into two components, one due to the environment and one due to space. There's a little space here in the Venn diagram to indicate that some environmental factors are spatially structured. What Ovaisky et al. Did was to add a third component, which is labeled I here, but C here, which is the co-distribution of species that cannot be explained by either E or S. And what we're proposing is that this is good, but then we can put individual species and individual sites in this space ECS. And what that means is that there are some, so if we think of these as species, there are some species that are just really, really good at tracking the environment. And that's what determines where they are. They're just, their distribution is very strongly constrained by the environment. Other species might not have any environmental sensitivity, and instead they might be very spatially structured. They're only found in clumps in the meta community, regardless of environments. Other species might be over here. They don't care about either environment or space, but their distributions is very much related to the distribution of other species, however that may be regulated. And we can try to predict the location of species. We can say the same thing about sites due to their attributes. So what do the results look like? In our simulations, we simulated this on landscapes, and we kind of did simple scenarios and then more complicated scenarios, etc. But what we found was that these types of analyses could be informative. So for example, if we had species, all the species have narrow niches, versus broad niches. If they have narrow niches, we find that they can be, that we get some species that are very strongly environmentally determined, whereas if we have broad niches, of course, they don't care. So no, we don't have that. We also have interaction among species, and that's true if we have broad niches as well. And the distribution of sites also follows a same overall pattern. So what that means is that there are some sites, like think of a lake here. This lake is occupied, by species that are predicted to be the competitive dominants. On the other hand, lakes that are over here don't have any competitive dominance. The species that are found in those lakes are just something else, maybe just due to chance or spatial patterning, or maybe avoiding other species that they don't like to compete with or something like that. So what I'm hoping you can see is that this gives us insights not just into the overall pattern, but into the dissection of the patterns according to the individual species that might be in different places in this graph, or individual lakes that are in different places in this graph. We can also play around with it a little bit more. So here we had a scenario in which some species were good dispersers, some species were poor dispersers, and they had narrow niches. And you can see that, just so I don't make a mistake here, that the species that are specialists, habitat specialists, are at the extremes of the gradient, are more strongly determined by the environmental conditions, but that this has a significant interaction with the dispersal abilities, with medium dispersal being over here and high and low dispersal being over here. And we can show that using these kinds of graphs. So here, dispersal has not very much effect on the environmental fraction in that graph. When we're looking at sites, at species that are habitat generalists, but a bigger effect when we're looking at species that are habitat specialists. We can also dissect by sites, and we find that this can be a very, very strong predictor. Lakes that are unusual would have very distinct species that are habitat specialists for those unusual lakes. So that's kind of cool. What about this covariation component, this C component? What does it look like? And how does it compare? So we think these are net interactions. How do they compare to the direct interactions? So we know what we put in. The direct interactions are shown here. And so the way we modeled this was for competition. And we just said that species that were in adjacent positions in their favorite environments competed strongly, and the other species did not. If we look at the net interactions among 5 different replicate runs, we find that we get something different. Okay, that was not unexpected, but we find that we have very low predictability in making these net interactions. And so that means that yes, the net interactions are present, but they are not really very easily predicted. And so, you know, if we invert these various matrices, they don't look like this. And so that's kind of disturbing. So maybe it's not so disturbing, because if you remember your matrix algebra, inverting a matrix is not a very linear type of mathematical transformation. And so, you know, it's going to be very sensitive to that. One other thing I want to say about this overall approach is that it really opens up a whole new way of thinking about the variation that we see within metacommunities. And, you know, I think Pedro will say a little bit more about this, but here's one thing that we just played around with a little bit in this one data set. We didn't use joint species distributions, we used something else, but it makes the point that if we predict the pure environment component in this data set of thousands of fit lakes in Canada fish communities, we find a typical result that we explain about 10% of the environment. If we look at pure space, we find that we get another 8% and another 8% if we look at the component that we just can't tell if it's environment or space. The apparent residuals, what we would have previously considered to be the stochastic component, is 75%. So this kind of is the usual sad story of community ecology, which is we get very excited about predicting very small components of community variation. And, you know, we usually say, well, that's okay. We're going to be happy with that because, you know, it's a tough job. You know, see if you can do better. Okay, let's see if we can do better. Here's the latent covariation in the residuals, 51%. What that means is that really, as far as we can tell, the actual unexplained stochastic variation is 25%. Holy whatever the right word is. I don't know what you say, but in polite society, but this is a very, very big difference. Okay, so we went from being confused about 75% of the variation to being less confused about 51%, with really only being totally confused about 25%. So this is a big difference. It means that our job as community ecologists has changed. Now we have to figure out what this means. And that's not going to be easy, but okay. It could be species interactions. It could be unmeasured environmental variables. It could be poorly specified spatial factors, or, you know, probably some other things. But, you know, now we have a target. So to give you know, this has all been pretty abstract. I wanted to kind of try to make it more tangible to lakes. These aren't always all very shallow, but that's okay. And it's part of a bigger project that I'm doing together with my postdoc, Priscila Engel. She's going to give a talk on some of this later today, I think. S19, is that today? I think so. And so we just calculated these same components for phytoplankton and zooplankton in these lakes. And what we find is that it seems like phytoplankton and zooplankton have relatively broad niches. There don't seem to be very many species that are very constrained environmentally. And so they're broad, I lost track of what I wanted to say. Broad niches. So on the environmental scale, the other thing that's surprising is that their distribution seems to be more strongly constrained by this co-distribution component than the environmental component. Okay, so maybe that's interactions, or maybe it's unmeasured environmental variables. The environmental variables that went in here were mostly water chemistry and physical variables. But maybe there's, I don't know, pollutants or something like that that is driving this pattern. We would not be able to tell. They also show quite strong spatial components. Maybe it's unmeasured spatial issues, or maybe it's dispersal effects. But it's a quite different picture than we might have expected. Okay. The other, if we look at the sites, so that previous graph was based on species. Here, each dot is a different lake. And the colors tell us which state they're in. And we were interested in this because we thought that, okay, maybe some parts of the US have lots of lakes that are highly connected spatially, and other parts are sparser. So what this shows is that, in fact, it kind of looks like something like that is going on. So Utah, Arizona, and Nevada don't have very many natural lakes. They're all the lakes that are in here are, not all of them, but the large majority are reservoirs, and they're more isolated to each other. Whereas other lakes seem to be a little bit more likely to have environmental components, especially Montana and California, which are much more likely to be natural lakes. We're kind of playing around with that idea. Friederike is going to talk a little bit more about that in her talk, so I'm just going to leave that as a look ahead. Okay, so I'm trying to convince you that this is progress, and here's my logic. The variation components are interpretable and informative. So, and the co-distribution, co-variation component is substantial in these 20 to 40%. We can study the internal structure, which we couldn't do before. We can do this for species, which people have suggested, especially the joint species distribution approach. And that's kind of considered to be the traditional, but we can also do this for sites. And that's cool because it allows us to merge meta-community ecology better with landscape ecology. The other conclusion is we're still very confused about indirect effects in these patterns. So one idea I had was that, okay, maybe we can't predict the actual shape of these net interactions or indirect effects very well, but maybe we can think of it as a statistical distribution. And that's inspired by, well, before I get to that, I did want to point out that last week, a paper came out in Tree by Bocciato et al. And they point out that joint species distribution models and stacked species distribution models are going to have a very hard time separating fundamental niches from realized niches, and that the co-distribution component is also likely to be involved in many other things. And so I don't know exactly what that means to anything I've said today, but I just thought it was important to point out that this paper is an important challenge to the some of these detailed conclusions. But the approach I'm looking to address is if we change our mindset and say, instead of trying to actually understand these net interactions, what if we think of them as statistical distributions of interactions? And it's inspired by work that these two guys did, Mathew and Guy Bunin. What we're working on right now is to link them to link their previous work on community assembly better to meta-community ecology, and to see how they might inform the way we understand these joint species distribution models. So here's what they do. So they have a distribution network, and they characterize the network. I thought I had a, let's go back over here, sorry. Their idea is that we have a very complicated set of things going on in diverse communities, but that they can be boiled down to these Locke-Volterra type equations. But we cannot really analyze these very easily, as we showed before, to get variables except through simulations and numerical solutions. And that's not very satisfying. What they suggest is that we can take a different approach based on statistical mechanics, which builds new equations that are based on kind of very aggregate properties of statistical distributions of species traits. And, and that these can give us predictions that are as good. And it seems like that's pretty good. Here, the lines is the prediction they get. The dots is the various simulations. You can see that they can predict total biomass pretty well. The fraction, the actual number of existing species pretty well. The variability through time, and the Shannon and the Simpson diversity index reasonably well. So that's pretty good. They also show that we can, it doesn't have to be completely disordered. So we can separate, for example, a food web into different trophic levels and characterize things. That will give different predictions, but we can generate those predictions. So it's a very flexible tool that's pretty cool. What we are working on now is to use this overall approach to study their effects on the output of Jsdms. And what we find is that if we know the environmental gradients and the species interactions, we find that the Jsdms do not do a very good job of identifying environmental filters. That confirms this proposition in this recent paper. We also find that if we know the direct interaction matrix, we can predict the co-distribution if we have no extinctions in the meta-community. That's not a surprise given the work by Bill Schaefer, but it does go against the conclusions in Boggiato et al. And if we can't predict the co-distributions, we find that we can predict their statistical features. I think I'm gonna skip over this a little bit just because it's a little bit messy. But we did that by comparing simulations where we had weak interactions and low dispersal with stronger interactions and high dispersal. Here, this matrix shows the distribution of each of 15 different, or whatever it is, however many species are in here, I can't remember. And you can see that they're quite different. Here is our predictions for the orange is the model parameters that determine the distribution of each of these species, and the blue is the estimate that we got from the JSDM. You can see that they don't do too good of a job here. They do a little bit better job here, still kind of problematic. But one of the other things to notice is that when we have these higher interactions, there are a number of species that go extinct in the meta-community. And that's going to change the nature of the net interactions. Here on this graph, it's kind of complicated. I'm going to focus just on this part. And it says, here's what the net interactions look like, or observed net interactions look like. Here's what the predicted net interactions. So if we have small interaction strength and low dispersal, we get very nice prediction. On the other hand, if the interactions are stronger and we have high dispersal, it's not so good. This is significant, but only because we have so many species. And if we forget about the details, but just say, what's the means under different scenarios, like high interactions versus low interactions, we find that we can predict these well using this method. And we can even predict the variance sort of well. I don't know what's up with this data point. I want to ignore it, but it's there, so can't ignore it. I have to do some more work. So it's an ongoing research program. I'm not providing any real answers, but I'm hoping to inspire you to study something along these lines. How can we test this? Well, we're going to have to compare the detailed model with the distribution model, the statistical mechanics probabilistic model. So that here's a little recipe for that. Get a bunch of species, estimate all pairwise interactions, estimate the net interactions by inverting the matrix, and see if this inversion corresponds to the prediction that we make using these statistical mechanics approaches. Where is this taking us? Well, I think we can go back to our little tinajas and say, what would I do today? I would be trying to predict the distribution of these correlations instead of the details. And so that's an important thing. I think if we can do this, we can expand questions to address how biodiversity affects ecosystem structure and other applied questions such as thinking about invasive species. When we have multiple invasive species, what are they doing? Well, they're changing the details of many things, which is the approach that most people take. But they're also changing the distribution of community parameters. And so that would change the way we ask this question. And that's all I have to say. I'm going to stop share and I'll take questions.
Thank you, Dr. Leibold, for a super interesting talk. We have 4 questions now. I think that's pretty much all we can take because of the time. So I'm going to start with the question by Stefano. He's saying, hi, Dr. Leibold, how to deal with metacommunity over time using data sets that were collected in several sites in monthly campaigns, for example?
Good question. So the joint species distribution models have been started to develop to look at time. I think the important thing to realize is that these methods are all in progress. So, you know, anything I say today is probably going to be outdated in the next hour when Pedro comes in. He will tell us, no, don't do it that way, do it this other way. And even if not, it will be outdated next week. So mostly, if you're thinking about, what I'm trying to get you to think about is thinking in this way rather than having specific recipe. There are a number of groups that are trying to address temporal change, not all of them through the joint species distribution approach. So, but most of the ones I know of don't focus on the internal structure part yet.
Thank you. The second question is by Ali. He's saying, thanks for a really nice talk. Is it possible to associate certain species traits with the relative location of species in the triangle model with space, environment, and other species?
In theory, yes. And in practice, yes. But maybe not as tightly as we could dream. But dreaming is important. So I think we can hope that, I'm using the word hope ironically because I know Pedro is going to use that word. We're friends, so we did not coordinate our talks, but I just guess that he's going to say. But yeah, that's the holy grail, is to be able to say, we now understand why that species is at the top of the triangle, why that other one is over here, and the other one is over here. We found that we could do that in these simulations to some degree.
Okay, thank you. Pedro, we already have a bunch of expectations about your talk.
You can thank me.
So the next question is by Celio Amorim. He's saying, hello, Mathew, congratulations on your nice talk. Could you please talk a little about assembly rules and how to reach comprehensive rules for communities or assemblages? Are null models always needed?
Thanks for the question. Yeah, I think I would like to see assembly rules be less about rules and more about process. And what I mean by that is, I think right now we have this filters kind of approach rather than thinking about the way these filters interact with each other. And the filters themselves, the way they're defined, ignores many processes. So they ignore things like, oftentimes they ignore disturbances. Another process that they ignore is in situ evolution. And so yes, it's possible to relate these findings to the idea of assembly rules, but I don't think that's, I think at some point we will find some limits to how useful that is.
The next question is by Nestor from Uruguay. He's saying, thanks for the interesting dissertation. The change in the type of interaction between two species, competition versus mutualism, depending on the context of the network, is it an aspect that we generally do not contemplate? Is it a frequent possibility?
I think that's a little bit open question, but I think we know enough. There have been enough studies done to know that we should expect this.
So another question by Bernadette. We try to look at large-scale multi-trophic co-response of food web components in Canadian lakes using MFA analysis and others. It has been published in Oikos. Is it a good avenue?
Bonjour, Bernadette. I know who you are. I'm not familiar exactly with what you're referring to, so I will need to read that more carefully to answer your question.
Shall we move on to the next one? Okay, so the next one is by Vera Hussar. Thanks, Matthew. Phytoplankton metacommunities used to show hundreds of species and about 2/3 are rare species. Does this approach work with so many species?
That's a good question. The approach we're doing here that I'm talking about is pretty closely related to the work that you did with Jamil. So I think I know where you are coming from on that front. The question of rare species is a really important one. Obviously, a single species, a species that's only found at one site somewhere is not very informative. The Jsdm people will tell us that they can use a Bayesian approach to parameterize their contribution. And I think this is an open question for statisticians. I'm not a statistician, but I know that it is a somewhat controversial claim. Maybe after Pedro's talk, you can ask him what he thinks about that question.
Back to you, Pedro. So another one by Monkey Chang. Hi, Matthew. I really enjoyed your talk. Thanks. Not sure whether this question can still be answered. Do you have experiences in using metacommunity theory to help improve water quality management?
Do I have experience? I have thought about it. I don't know if that's called experience. Yeah, so if you go to Frédéric's talk, you might find that interesting. You'll be disappointed because our answer is not very conclusive and we are working on it some more. But the idea is that if we can manipulate dispersal, we might find ways that give different water quality control on things like algal blooms and stuff like that. So if like one way to think of it in practical terms is it seems like the most reliable species to control algae is Daphnia, regardless of species. So Daphnia are the super controllers. It's not super clear why that is, but they are the most reliable indicator of algal control. But Daphnia can be very strongly dispersal limited and also environmentally constrained by, for example, fishes or inadequate refuge from predation. And so manipulating those things, changing the environmental parameter or the dispersal parameter could be important.
So I think I'll take these two final questions to give Pedro time to settle in and adjust. So the next one is Nicholas Vidal. Thanks for your talk. I want to know how the dispersion in time diapause, for example, can affect the approach.
Thank you again. So diapause can be thought of as dispersal through time. And that's an interesting way to think about it. It's a little bit more complicated than that. There's a paper that I'm a co-author on by Nathan Wisnowski, where we looked at dormancy in metacommunities. And the effect of dormancy of diapause depends on whether, or can depend on whether whether diapause is positively or negatively correlated with dispersal. And different groups of organisms seems to have different correlations. So in zooplankton, usually the resting stages are also dispersal stages, and so they tend to be positively correlated. But in many terrestrial plants, seeds that are highly, that have high dormancy are bigger and do not disperse as far. And so there's a negative correlation there. So the answer is it matters, but it's more complicated than just yes or no.
So the final question by Antonio Jimenez. Have you measured the effect of cryptic species or the importance of different resolution in the taxonomy? And what application does this approach, the Jsdm, have?
Cryptic species are wonderful challenges to our paradigms. So cryptic species that are strongly allopatric are not very surprising. What is surprising is when we find cryptic species that are strongly associated with coexistence. And the most obvious explanation for such coexistence is that they coexist through neutral, equivalence, kind of like Hubble's theories of metacommunity ecology. And I think there's some examples of that. I have a student who did a dissertation on amphipods in Hyalella, and she looked at whether they showed any niche differentiations at the local scale, and found that, so there are 3 species that are found ubiquitously throughout the Midwestern US, and they coexist ubiquitously. 2 of them seem to be equivalent species as far as she could tell. And the 3rd one had a small niche difference that could explain its coexistence. So yeah, I think they reveal much more fine scale sort of niche dynamics than we might have expected.
So thank you, Matthew. I think we should end here, but just so you guys know, Matthew is going to be with us this afternoon again in our early career researcher workshop. Of course, he's not going to talk about metacommunity, that's not the intention, but we have some time in the end. So if you still want to catch up, or if you have more questions, maybe there's going to be some time at the end for that. But please join us in the afternoon again.
Don't forget, we are a meta community.
Exactly. So thanks a lot, Matthew. And that's it for now.